Okay, good afternoon, everyone. So I'm Aiala Salvador, and I will present my work that has been carried out at the University of the Basque Country and in collaboration with a research group from the NIH at the US. This work is entitled Improved Adjuvants for Vaccine Development, Surface and Adjuvant Modified PLGA Microspheres. Excuse me. So, for the development of vaccines, the most traditional way was to, or has been, the preparation of inactivated vaccines, but they used to have several drawbacks, especially related to the safety. So nowadays, the most research have been conducted by using antigens that are subunits, like peptides and proteins. However, it's quite difficult to achieve a high immune response by using just such a small molecule like a peptide or a protein. And they are very unstable, especially because they are degraded by proteases. So one of the alternatives in order to improve their stability is to include them into a drug delivery system. So these systems protect the peptides or the proteins or the antigens, and they have other advantages. They can produce a controlled release from the antigen. They can also produce a depot effect at the site of injection from which the antigen can be released by a controlled manner. They can improve also the absorption and also the bioavailability of these molecules. We can also target specific cell types. In this case, they will be immune cells. And the last advantage, among others, or among the most important ones, it's that the delivery systems allow the incorporation of more than one molecule into the same system. And we'll see why this can be an advantage. Well, among the several possibilities among the drug delivery systems, for our work, we have chosen polymeric particles. In our case, there are polymeric microparticles made of this polymer, which is called PLGA. This is a copolymer made of lactic and glycolic acid, so that's why it takes the name, polylactic coglycolic acid, so PLGA. It's quite interesting because it's biodegradable and biocompatible, and it has been approved by the most important regulatory agencies like the FDA from the US, the Food and Drug Administration, and the European Medicine of Agency for its biological use. So this is very briefly the mechanism that this particle follow once they are injected for vaccination purposes. So we incorporate our antigens into the microparticles, and then the aim is to target the antigen-presenting cells, in this case, the dendritic cells. These cells are very important to start or to trigger an immune response. Depending on how the interaction and the activation of these dendritic cells is, we can activate one arm or another from the immune response. So, very briefly, if these particles are able to release the antigens and present them into major histocompatibility molecules class 1, we can trigger or we can start a cellular immune response with the activation of cytolytic T lymphocytes. Instead, if the particles release the antigen, and this is presented into major histocompatibility molecules class 2. We activate the Th2 or CD4 T cells. We activate macrophages, B cells. It starts the secretion of antibodies. So we activate the humoral arm of the immune response. So depending on the type of vaccine that we want to prepare, depending on the pathogen, we have to focus to one or another arm of the immune response, and sometimes it is interesting to activate both, like, for example, in the case of diseases like cancer or maybe malaria or diseases caused by viruses. Which are some drawbacks from this system, that sometimes just with the delivery system and the antigen, it's still difficult to produce a strong immune response or a long-lasting one. So what can we do? We can incorporate an adjuvant, which is a molecule that also stimulates the immune response, in the same system, because we can incorporate in the same particle the antigen and other molecules, and in this case, the adjuvants. Which can be the mechanism of action? The depot effect produced by the delivery system itself that can target the cells, and inclusion of other immunostimulatory adjuvants. So taking all of this into account, the objectives of this work were to first in vitro evaluate these PLGA microspheres and several modifications that we perform on them, and to evaluate if those modifications had any influence on the uptake by the monocytes. Then, second, to in vivo evaluate the uptake by the immune response produced by the microspheres using a model antigen, which is BSA, bovine serum albumin. And third, to determine if the inclusion of immunostimulatory adjuvants into the same microspheres with the BSA has a significant effect on the immune response. So first, we modified the plain PLGA microspheres with this polymer, which is called PEI, or polyethylenimine, in order to modify the surface charge of the particles and to become it cationic. This is supposed to be an advantage for targeting the cells because the cell surface, as you may know, it's negatively charged. So if we produce cationic particles, we can improve the interaction by the electrostatic charges. So here you can see scan electron microphotographs. These are the unmodified particles, and here we have the PLGA modified particles. And as you can see, there is no difference, or there are not big differences, nor in the size, in the morphology, etc. We also carried out several other characterizations, and we reached to the conclusion that the modification or the incorporation of this polymer did not significantly modify the structure of our microparticles. Next, we evaluated if freshly isolated human primary dendritic cells and monocytes were able to take up our microparticles, which is a very important fact that has to be developed for an immune activation. So we compared our unmodified microspheres and our cationic microspheres, and as you can see here, these ones are the unmodified one, and these ones are the cationic ones. The uptake was significantly enhanced for all the cell types, for the monocytes and the two main dendritic cell subsets, which are myeloid dendritic cells and plasmacytoid dendritic cells. Then we divided, or we also measured the uptake by the different myeloid dendritic cell subsets, because they have different functions, and we found that all of them were also able to take up our microspheres, microparticles. This is very important because each subset has a different function. So if we are able to activate all of these dendritic cell subsets, that can mean that we can be able to activate a stronger response. And here you can see an example of a confocal microscopy. These are monocytes, and here you have the myeloid dendritic cells, and here these green spots are the microparticles that have been internalized into the cells. Then we tried to do something similar, but in this case in vivo. So for that purpose, we took some valve C mice, and we administered our particles intradermally. And after 48 hours, we removed the lymph nodes and the spleens, and we tried to measure the presence of the microparticles. Sadly, we were not able to detect any fluorescent signal, nor in the spleens, nor in the lymph nodes, but we could appreciate that the lymph nodes from mice receiving the microspheres were higher in size in comparison to the mice receiving a negative control, which was PVS. In addition, we found that after those 2 days, there was still a high skin depot compressing the microparticles. And after analyzing this tissue, we found that it was very, very positive for the microparticles. So we believe that probably the microparticles are here, and they are being released very slowly to the lymph nodes. And probably if they are traveling inside the immune cells, they can be inside the lysosomes, and the fluorescence can be losing in that way. Then we use the microparticles comprising the mobile antigen VSA, and we immunized these valve C mice, and we measured the antibody production during 12 weeks. In addition, as we know that the immunostimulatory adjuvants can improve even more the immune response, we decided to include several combinations of the adjuvants into the formulation, into the particles. So we used MPLA, which is monophosphoryl lipid A, which is a TLR4 agonist, poly-IC, polynosinic polycytilic acid, which is a TLR3 activator, and alpha-galser, which is an activator of the natural killer T cells. So what we find here is that the adjuvant combination, it does not have a very big influence, and the higher differences are found depending on the surface charge of the particles. So when we used our cationic or modified microparticles, the immune response was higher in most of the cases in comparison to the plain microparticles. Here we have this Freund's complete adjuvant, which is a positive control, and for most of the time points, the particles with the cationic surface charge produced very similar antibody levels in comparison to the positive control. Next, we decided to evaluate the ability of the microspheres to trigger different antibody subclasses. This IgG2A in the mice it's released when a Th1 immune response is activated, and the IgG1 is activated when a Th2 immune response is activated. So sadly, we could not detect any differences in all the microparticles. The only difference is when we compare it to the positive control. So that means that even though the total amount of antibodies is very similar, the particles are able to activate more than one arm of the immune response in comparison to the positive control. And lastly, we decided to ex vivo re-stimulate the splenocytes and to measure the cytokines that they were able to release. So for that purpose, 15 weeks after immunization, we take off the spleens and we isolated the splenocytes, and we put them in contact with the antigen. And after 48 hours, we measured the production of interferon gamma, interleukin 4, and interleukin 17. Interestingly, depending on the surface charge, the cytokine profile was different. So for the particles that have a positive surface charge, the interferon gamma production was higher, and they produce very few IL-4 and IL-17. This is indicative of a Th1 activation. In contrast, the non-modified particles were able to trigger IL-4 and IL-17 cytokine levels, and that means that they can activate a or they are activating a Th2 arm of the immune response. So from all of this, we can conclude that all the dendritic cell subsets, as well as monocytes, are able to internalize our PLGA microspheres. This uptake varies with the nature of the particles. Third, that all the microspheres can efficiently prime antibody responses and also T-cell responses with a lower influence of the adjuvant combination. The higher influence, it's the one from the surface charge. And these surface charges seem to have an effect on the skewed arm of the immune response. So here is my research group from the University of the Basque Country and collaborators from the NIH. And thank you very much for your attention.
Any question, please?
Thank you for your valuable lecture. I would like to ask, I know that antigen engulfed by macrophage, any protein, any antigen, any antigen engulfed by the macrophage for starting the processing of immune stimulation. Here, you concentrate on the microsphere uptake by the immune cell. Where is the role of antigen here? What is the role of this particle? I expect this particle with the negative charge to combine with the protein to be engulfed by macrophage.
Behavior of the non-modified microparticles. And in this one, what we did was to compare first in vitro both, and then just in vivo the second ones, the modified ones. So we know that the unmodified ones are also quite good for producing an immune response in vivo. But it was not enough. So that's why we decided to do or to carry out this second project.
No, what I meant is the key step in of antigen to the macrophage is uptake of antigen by macrophage. What is the role of negative sphere here in this process? For instance, usually we use, for example, in experimental animal, an adjuvant known as complete Freund adjuvant. This adjuvant compared of oil and dead microbacterium, for example.
Yeah.
So the cell wall of microbacterium recruits macrophage to the area of antigen to allow for macrophage to engulf the antigen. What is the role of this microsphere?
Oh, yeah. So, the thing is that as you increase the size of the antigen, it resembles more to a pathogen. And then it has also some importance, the site of injection. In this case, we have chosen the skin because there are a lot of dendritic cells. So the idea, or our aim, is not only to target macrophages, because otherwise it's quite difficult to stimulate Th1 or cytolytic or CD4, CD8 activation. So what we wanted to know in the first part was if we were able to target not just the macrophages, but also other dendritic cells. So we think that we have seen that we can target, of course, the macrophages were the cells with the highest uptake, but even the PDCs that are not phagocytic cells could take up the particles. Of course, that happens in vitro. Probably in vivo, it will be different. But if you administer the particles in a site where they can be in contact with dendritic cells, like in the skin, where you have Langerhans cells and dermal dendritic cells, you are increasing the chance of being taken up by those dendritic cells, and then they migrate to the lymph nodes.